the coelacanth. A fisherman hauls in his net off the South African coast on December 22, 1938, and pulls up something that stops the world. Not a new species, not a rare catch. A fish that every scientist on Earth agreed had been dead for 66 million years. The last time this animal was alive, Tyrannosaurus rex still had 10 million years left to run. Curator Marjorie Courtenay-Latimer notices it on the dock, sketches it in a panic, and mails the drawing to ichthyologist J.L.B. Smith. His response, now legendary, was reportedly this: he would not have been more surprised if he had seen a living dinosaur. This is the coelacanth, and its existence is one of the cleanest violations of what we think we know about extinction. The coelacanth lineage first appears in the fossil record approximately 420 million years ago, long before the first dinosaur, long before the first flower, long before anything walked on land on four legs. It cruised through the Devonian, the Permian, the Triassic, the Jurassic. Then, 66 million years ago, the Chicxulub impactor hit the Yucatan Peninsula with energy equivalent to roughly 1 billion atomic bombs. It triggered global firestorms. It threw enough debris into the atmosphere to block the sun for a decade. It collapsed the food chain from the top down and the bottom up simultaneously, killing 75% of all species on Earth, including every non-avian dinosaur that had ever drawn breath. And the coelacanth vanished from the fossil record that same day. Gone. Not a single fossil trace for 66 million years across 6 entire geological epochs. And then Marjorie Courtenay-Latimer opened that crate on a South African dock. The shock is not just that it survived, it is how completely it failed to change. A genomic study published in Nature in 2013, led by researcher Chris Amamiya and colleagues, confirmed that the coelacanth has one of the slowest molecular evolutionary rates ever recorded in any vertebrate. Its genome is, in scientific terms, nearly frozen. The living animal is so anatomically close to its 300 million-year-old fossil relatives that the two are described as almost identical. In 2013, that is not a poetic comparison. It is a measured molecular one. And then there are the fins. The coelacanth's paired pectoral and pelvic fins are lobed and fleshy, each containing an internal bone structure that mirrors the layout of a tetrapod limb. One bone, two bones, many bones. The fins move in an alternating gait, like legs, like walking. When the coelacanth swims slowly in the deep, it moves those fins the way a land animal moves its limbs, left front with right rear, right front with left rear. We are watching, in real time, the motion that eventually put the first creature on land, still being performed by an animal that never made that trip. In its snout sits a rostral organ that detects weak electric fields, a sensory system found in no other living fish except sharks and rays, suggesting this animal carries technology assembled before modern fish anatomy was even standardized. A second population was discovered in 1997, not off Africa, but off the coast of Sulawesi, Indonesia, separated from the African coelacanth by the entire Indian Ocean. Marine biologist Mark Erdman found it in a local fish market. A separate species, Latimeria menadowensis, living on the other side of the world, in a range the fossil record gives us almost no evidence for. 2 populations, 2 oceans, 66 million years of silence. The mainstream scientific explanation is the deep-sea refuge hypothesis. The coelacanth survived because it occupied cold, deep water that was partially shielded from the catastrophic surface conditions of the KPG impact. The absence from the fossil record is explained by simple geology. Deep-sea organisms rarely fossilize. Their remains decay into sediment rather than being preserved in rock we can access. The slow evolution reflects a stable, unchanging environment that demanded no adaptation. It is a reasonable explanation presented by serious researchers. But it runs into a quiet problem. Every other large vertebrate group that survived the KPG event left a continuous fossil record. Crocodilians left one. Turtles left one. Birds left one. The coelacanth left nothing across 66 million years, even though it was apparently living in ocean zones that do occasionally yield fossils from other species. And its molecular evolution rate, according to the 2013 Nature study, is statistically anomalous even compared to other deep-sea animals living in the same supposedly stable environment. Other species in the same depths evolved at normal rates. The coelacanth did not. Some researchers ask whether the near-frozen genome is a clue rather than a footnote. What biological mechanism allows an animal to essentially pause its own evolution across timescales that reshape continents? Could the rostral organ, that electric field detector with no modern analog, represent sensory architecture from an older template of vertebrate life, one we haven't fully characterized? And if a large distinctive electric blue fish hid from science until 1938, what else might still be hiding in those same depths, waiting for someone to open the right crate? The coelacanth survived one extinction event. The creature we turn to next survived 4. The horseshoe crab. Picture the Delaware Bay on a warm spring night. The tide is coming in. And moving through the surf, in their hundreds of thousands, are armored creatures that look like they crawled off a medieval woodcut. Brown, dome-shelled, trailing a rigid spike of a tail. They drag themselves up the sand in the dark, as they have done every spring for nearly 500 million years. 4 mass extinctions have passed since the first of these animals ever existed. They watched the trilobites vanish. They watched the dinosaurs rise and fall. They watched the mammals inherit the earth. They are still here, climbing the same beaches, and their blood is currently sitting in a medical laboratory somewhere, testing whether the vaccine in your arm is safe to inject. This is the horseshoe crab. And its existence is not just a mystery of survival, it is a mystery of purpose. The oldest confirmed horseshoe crab fossil, a species called Lunataspis aurora, was described by researchers David Rudkin and Graham Young in a 2008 paper in the journal Paleontology, dated to approximately 445 million years ago, found in what is now Manitoba, Canada. Look at that fossil and then look at a living horseshoe crab. The same 5-part body plan, the same compound eyes, the same telson spine dragging behind. 445 million years of time, and nothing of consequence changed. In that span, the horseshoe crab survived the Ordovician extinction, roughly 445 million years ago, which killed approximately 85% of all species on Earth. It survived the late Devonian extinction, roughly 375 million years ago, which killed approximately 75%. It survived the Permian extinction, the Great Dying, 252 million years ago, the worst catastrophe in the history of life, triggered by the Siberian Traps volcanic eruption, which raised ocean temperatures by an estimated 10 degrees Celsius, acidified the seas, and eliminated 96% of all marine species. 96%. And then it survived the Cretaceous Paleogene extinction 66 million years ago that killed the coelacanth's fossil record and every non-avian dinosaur alive. 4 separate apocalypses, 4 separate mechanisms, cold and heat and acid and impact, and the horseshoe crab walked through all of them without changing its body plan in any meaningful way. And then there is the blood. In 1956, Frederick Bang, a researcher at the Marine Biological Laboratory in Woods Hole, Massachusetts, discovered that horseshoe crab blood contains a substance called limulus amebocyte lysate, or LAL, which clots instantaneously around bacterial endotoxins. Not around the bacteria themselves, but around the molecular debris they leave behind at concentrations as low as 1 part per trillion. That sensitivity has no pharmaceutical equivalent. We have not built anything that matches it. As a result, every intravenous drug, every implanted medical device, every vaccine produced today is tested for bacterial contamination using LAL extracted from living horseshoe crabs. The blood is literally blue, copper-based rather than iron-based, a chemical inheritance from ancient marine chemistry. The United States pharmaceutical industry harvests approximately 500,000 horseshoe crabs per year for this purpose. The crab's visual system is equally strange. It has 5 pairs of eyes, including compound lateral eyes that have been studied more extensively than almost any other visual apparatus in neuroscience. Researcher H. Keffer Hartline won the Nobel Prize in Physiology or Medicine in 1967, partly through his work on the horseshoe crab's eye. The animal effectively became a cornerstone of how we understand vision itself. The standard explanation for this survival record is ecological flexibility. The horseshoe crab tolerates a wide range of salinity and temperature. It eats mollusks, worms, and algae. Its eggs are a critical food source for migratory shorebirds, suggesting deep integration into ecosystems that survived each extinction. Its simple body plan required fewer specialized adaptations to maintain, and its burrowing behavior may have sheltered it from surface-level catastrophes. These are serious arguments made by serious scientists. But simplicity and flexibility describe dozens of other Ordovician arthropods, and they are all gone. Trilobites were flexible. Eurypterids were flexible. The seas were full of ancient arthropods that survived 1 or 2 extinction events and then vanished in the 3rd. The horseshoe crab survived them all and emerged unchanged, each time carrying in its blood a detection system so precise that modern medicine has built its safety infrastructure around it. Some researchers quietly ask whether the LAL system is a window into a marine bacterial environment radically different from today's, one that demanded biological sensors of extraordinary sensitivity, sensitivity we are only now beginning to understand the implications of. If so, the horseshoe crab isn't just a survivor, it is a message from the conditions of an ancient ocean, still functional, still readable, still keeping people alive. From the floor of the Ordovician Ocean, we travel to the shallow coastal waters of Western Australia, where something even older is still breathing. The stromatolites. You are standing at the edge of a shallow lagoon in Shark Bay, Western Australia. The water is warm, hypersaline, roughly twice the salinity of normal ocean water. Jutting from the shallows are rounded, lumpy gray formations that look like rocks somebody stacked by hand and then abandoned. They are not rocks. Each one is a living colony of cyanobacteria, building itself grain by grain, layer by microscopic layer, at a rate of approximately 0.3 millimeters per year. A stromatolite 1 meter tall represents roughly 3,300 years of biological accumulation. And the organism doing the building has been doing exactly this in exactly this way for 3.5 billion years. To put that number in context, the horseshoe crab, which we just called ancient, has existed for less than 13% of the time stromatolites have been alive. The coelacanth lineage, ancient at 420 million years, represents less than 12% of the stromatolite story. These organisms were here before complex cells existed, before animals existed, before the concept of a spine had been invented. They are not survivors of extinction events. They preceded them by so long that the word survivor barely applies. The scientific identification of fossil stromatolites as biological in origin was confirmed in a landmark 1993 study by researcher J. William Schaff, published in Science, which dated microfossils from the apex chert of Western Australia's Pilbara region to approximately 3,465,000,000 years ago. More recent analysis by Nora Nofke and colleagues, published in Astrobiology in 2013, dated stromatolites from the Dresser Formation in the same Pilbara region to 3,480,000,000 years ago. The living specimens at Hamlin Pool, Shark Bay, were designated a UNESCO World Heritage Site in 1991. The site covers approximately 135 square kilometers. But the fact that makes stromatolites genuinely extraordinary is not how old they are. It is what they did. Approximately 2.5 billion years ago, the cyanobacteria building these structures triggered an event called the Great Oxidation Event. They had been photosynthesizing, consuming carbon dioxide, and releasing oxygen as a waste product for roughly 1 billion years by that point. And eventually, they produced enough oxygen to fundamentally alter the chemistry of Earth's atmosphere, raising free oxygen from near zero to detectable levels for the first time in the planet's history. This transformation was itself the largest mass extinction event in Earth's history. It killed the anaerobic organisms that had previously dominated the planet, organisms for whom oxygen was a poison. The cyanobacteria that caused that extinction are the same organisms building those formations in Shark Bay today. In other words, stromatolites did not just survive mass extinctions. They caused one, and then they survived 5 more. The deepest impossibility here is not their longevity. It is their stillness. Under electron microscopy, living Shark Bay cyanobacteria appear morphologically identical to their 3.5 billion-year-old fossil relatives. Not similar. Identical. 3.5 billion years of Earth history, which includes the formation of the Himalayas, the opening and closing of multiple oceans, 5 mass extinctions, the entire evolution of every animal that has ever lived, and these organisms registered no detectable pressure to change their form. Microbiologists explain this through the concept of irreducible minimalism. Cyanobacteria are prokaryotes, cells without a nucleus, representing one of the most chemically efficient life forms possible. They require only sunlight, water, and carbon dioxide. Because they need almost nothing, there is almost nothing that can eliminate them, and almost no environmental pressure sufficient to demand they become something else. It is a compelling explanation, but it sidesteps the most philosophically unsettling detail. The cyanobacteria living in Shark Bay are not merely biochemically similar to their Archean ancestors. They appear, as far as we can measure, to be the same thing. Not a descendant. Not an evolved version. The same template still running on a planet that has been transformed beyond recognition multiple times since that template was written. NASA's Mars exploration program has identified potential biosignatures in rock formations at Jezero Crater that structurally resemble stromatolites. The living specimens at Shark Bay are, as a result, the closest analog we have to what life on another world might look like. Some astrobiologists ask whether the cyanobacterial design represents something like a biological floor, a minimum viable architecture for life that, once achieved, cannot be improved upon and therefore does not need to be. If that floor was reached 3.5 billion years ago on Earth, the question of how life first assembled that template remains as open as it has ever been. From something nearly invisible, building the world grain by grain, we move to an island at the edge of the world, where the last survivor of an entire reptile order is staring at you with 3 eyes. The tuatara. It looks like a lizard. Every instinct you have says lizard. Greenish-gray, scaly, roughly 60 centimeters long, sitting very still on a rock on a small island off the coast of New Zealand. But the moment you look at the top of its skull, just behind its eyes, at the pale, scale-covered disc sitting there like a third socket, you realize you are not looking at a lizard at all. You are looking at the last living member of an entire reptile order that flourished when the dinosaurs were young, diversified across every continent on Earth, filled the Jurassic and Cretaceous with dozens of species, and then collapsed everywhere in the world except one set of small islands at the bottom of the Pacific. And the organ on its skull, that pale disc with a lens and a rudimentary retina wired directly to its brain, is something no lizard has ever possessed. The Tuatara's lineage, the order Rhencocephalia, first diverged from all other reptiles approximately 240 million years ago in the Middle Triassic, according to molecular clock estimates published by researcher Mark Jones and colleagues. At its peak, the order contained over 40 known species distributed across multiple continents. It was a successful, globally distributed reptile group living alongside early dinosaurs, thriving through the Jurassic, still present in the Cretaceous. Then, in the aftermath of the KPG extinction, as mammals and lizards expanded into every available ecological niche, the rhynchocephalians were systematically replaced. Everywhere except New Zealand. New Zealand separated from the supercontinent Gondwana approximately 80 million years ago, before placental mammals ever evolved. The Tuatara survived because it never met the predators that killed everything else like it. The result is not a lizard, it is a phylogenetic ghost, the sole representative of a vertebrate order as distinct from lizards as lizards are from crocodiles. Finding a Tuatara is the biological equivalent of finding a living animal as evolutionarily separate from everything around it as a dolphin is from a wolf. Except the Tuatara has been separated for 240 million years. Its biology is full of surprises. Tuatara remain active at body temperatures as low as 7 degrees Celsius, making them one of the only reptiles functional in near-freezing conditions. Most reptiles would be comatose at that temperature. They grow continuously for approximately 35 years before reaching full size. They can live past 100 years in captivity. Females reproduce only once every 4 years. Their teeth are not replaced. They are fused directly to the jawbone and wear down over a lifetime until elderly Tuatara are chewing with bare bone, a feeding mechanism with no equivalent in any living reptile. Males transfer sperm without a penis, using a method shared with some birds and considered anatomically primitive among reptiles. And then there is that third eye. The parietal eye on the top of the skull is fully formed in juvenile Tuatara, visible as a distinct structure with a lens, a rudimentary retina, and a direct connection to the pineal gland in the brain. In adults, it becomes covered by scales and apparently loses direct visual function, though the pineal connection remains active in regulating circadian rhythms and seasonal behavior. The standard explanation is that this organ tracks changes in day length, helping the Tuatara calibrate its internal biological clock to seasons and temperature cycles. That explanation is scientifically reasonable. But the parietal eye did not evolve in New Zealand. The Tuatara inherited it from an ancestor 240 million years old, which means this organ was present and presumably functional in every rhynchocephalian that ever lived. 40 species across multiple continents, all carrying a photosensitive organ on top of their skulls for 240 million years. What was it doing for those 240 million years that made it worth keeping? Seasonal light detection is a plausible, partial answer. But some herpetologists have proposed that in early rhynchocephalians, when the organ was younger and perhaps less covered, it may have served a more active navigational function, something closer to solar tracking, something more directional. The connection to the pineal gland, the same gland referenced in ancient traditions across cultures as the seat of inner perception in a 240-million-year-old reptile lineage, raises questions that anatomy alone does not settle. By 1900, introduced rats and mustelids had eliminated tuatara from mainland New Zealand entirely. The species now survives on 32 offshore island sanctuaries. A population that watched dinosaurs come and go was nearly finished off by rats. From a remote island where a survivor hides in plain sight, we move to a hidden canyon in Australia, where a tree kept its secret for 2 million years. The Wollemi Pine. On September 10, 1994, a park ranger named David Noble rappelled into an unmarked canyon in the Blue Mountains of New South Wales, Australia, and landed next to a tree his brain could not process. He was an experienced field officer. He knew Australian flora. But this tree, tall and dark with extraordinary bubbly bark unlike anything in any reference book he had ever used, was simply not in the catalog. He collected samples. Botanist Ken Hill at the Royal Botanic Gardens in Sydney identified them and had to read the analysis twice. The tree was a Wollemi pine, a species known only from fossils, last recorded in the fossil record approximately 2 million years ago. Noble had rappelled into a living museum, and the location of that canyon, to this day, is classified information. Pollen morphologically identical to Wollemia nobilis appears in Australian rock strata dated to at least 90 million years ago, deep in the Cretaceous period, 24 million years before the asteroid that killed the dinosaurs. The species survived that extinction and kept going through the Paleocene, the Eocene, the Oligocene, the Miocene, then vanished from the fossil record approximately 2 million years ago during the Pliocene as the climate dried and cooled across Australia. The trees in Noble's Canyon had, by every measure of the fossil record, been gone for 2 million years. And yet there they were, reproductively active, structurally normal, growing in a gorge so narrow and so deep that aerial surveys had simply missed them. The formal description of the species, published in the journal Telopeia in 1994 by Hill Noble and colleagues, named it Wollemia nobilis in honor of the man who found it. The trees can reach heights exceeding 40 meters. The bark, described as brown and bubbly, is found in no other known plant species. Subsequent surveys located fewer than 100 mature adult trees distributed across fewer than 5 known sites, all within the same narrow canyon system in Wollemi National Park, approximately 150 kilometers northwest of Sydney. Then came the genetic analysis, and it delivered the deepest shock of all. Every wild Wollemi pine shows virtually zero genetic diversity. Not low diversity, near zero. Every tree in that canyon is genetically almost identical to every other tree. In evolutionary terms, this means the population has been reproducing from an extremely small gene pool, perhaps a single lineage, for an extraordinarily long period of time. A tree that shared a world with Cretaceous dinosaurs had been, for lack of a better word, cloning itself in a hidden canyon for longer than modern humans have walked the earth. Standard population genetics has a clear prediction for what happens to populations this small and this inbred. They collapse. Inbreeding depression accumulates genetic defects across generations, reproductive success declines, and within thousands of generations, extinction follows. The Wollemi pine does not appear to have read that model. The trees are healthy. They reproduce normally. They show no obvious signs of the genetic dysfunction that should, by every textbook measure, have ended this lineage long ago. Botanists offer two explanations. First, the canyon refugium itself. The deep, humid gorge created a microclimate buffered from the Ice Age conditions that dried and cooled the surrounding landscape, allowing a small population to persist where nothing else of its kind could. Second, and more intriguing to some researchers, a hypothesis involving deep biochemical stability. The Wollemi pine may carry a suite of chemical defenses and developmental programs so thoroughly conserved across 90 million years that the tree simply does not need genetic variation to respond to stress. Its responses are already encoded. Its toolkit is already complete. It does not need to improvise. Since the discovery, propagation programs have distributed Wollemi pines to botanic gardens in over 50 countries. The tree that hid for 2 million years is now sold in garden centers. That detail alone carries a strange weight. Something ancient enough to have existed when Gondwana was still assembling itself is now available at a nursery. Some botanists have quietly asked how many other Cretaceous plant lineages might be hiding in comparable canyon systems elsewhere in Australia, in the Amazon, in the Congo, in the Himalayas, in places no one has yet rappelled into. If a tree 40 meters tall and unlike anything in any reference book could hide in a gorge 150 kilometers from Sydney until 1994, the question of what else we haven't found yet is not a rhetorical one. A hidden tree is one thing. What we turn to next is harder to dismiss because it has been hunting in the open ocean since before the first flower ever bloomed, and almost no one has ever seen it alive. The frilled shark. In January 2007, staff at the Owashima Marine Park in Shizuoka, Japan, noticed something tangled near the surface in a fisherman's net. They brought it in and filmed it moving through a holding tank. What the camera captured does not look like a shark. It is approximately 2 meters long, eel-like, moving in slow undulations. Its 6 gill slits are fringed with ruffled filaments that ripple as it breathes, like the collar of a Victorian dress. Its jaw opens wider than seems mechanically possible, nearly 180 degrees, a dark funnel ringed with 300 curved, trident-shaped teeth arranged in 25 rows. Its eyes have never adapted to surface light. The animal died within hours of capture. It was the first time a living, behaving frilled shark had ever been filmed. The frilled shark, Chlamydocellus anguineus, was first formally described by American ichthyologist Samuel Garman in 1884 from a specimen collected off Japan. Its fossil ancestors appear in the record approximately 80 million years ago, in the late Cretaceous period. The great white shark evolved its current form roughly 11 million years ago. The hammerhead, the bull shark, the tiger shark, all of them are geologically recent, shaped by forces that arose after the dinosaurs were already gone. The frilled shark's skeletal structure, jaw articulation, and tooth geometry appear in the fossil record 80 million years ago and have not meaningfully changed since. Anatomist Carl Gegenbauer noted in the 19th century that the frilled shark's body plan is more primitive than any other living shark species, retaining features that modern sharks discarded tens of millions of years ago. The 6 gill slits are the ancient condition. Modern sharks standardized on 5. The terminal mouth, positioned at the very front of the head rather than underneath, is another primitive retention. The frilled shark looks like a diagram of what sharks were before sharks became what we think of as sharks. But the deepest impossibility in its biology is not anatomical. It is reproductive. The frilled shark's gestation period is estimated at approximately 42 months, 3.5 years, based on embryo development studies from deep-water trawl specimens. 3.5 years. That is longer than an elephant, longer than a sperm whale, longer than any other vertebrate whose gestation has been reliably measured. The embryos develop inside the mother with no placental connection. They are sustained entirely by yolk. At near-freezing deep water temperatures, metabolism slows to a crawl, and development proceeds at a rate that would be incomprehensible by surface standards. The animal lives at depths between roughly 120 and 1,280 meters in cold, low-oxygen water that has changed very little since the Cretaceous. Its liver makes up an extraordinary proportion of its total body weight, providing the buoyancy and energy storage necessary to sustain a multi-year pregnancy in an environment where prey is sparse and encounters are rare. As of the time of writing, fewer than 100 frilled shark sightings have been formally documented worldwide. Its behavior in the wild remains almost entirely unknown. We have no reliable data on how it hunts, how it selects mates, or how it navigates across the deep Atlantic and Pacific where separate populations appear to exist. The mainstream explanation is the deep-sea stability hypothesis. Cold, stable, nutrient-poor environments select for slow metabolisms, low reproductive rates, and morphological conservatism. The frilled shark's 3.5-year gestation and its Cretaceous body plan are both products of a habitat that demanded nothing fast and nothing new. It is a logical argument, but there is something in the anatomy that does not fit. The near 180-degree jaw opening is not a primitive retained feature. It is a derived, specialized structure found in no Cretaceous frilled shark fossil. This supposedly frozen animal evolved something. It evolved one thing. It evolved a better way to swallow prey whole. Which means the frilled shark is not simply an organism that failed to update. It is an organism that chose its one update carefully and left everything else alone. Some paleontologists have proposed that the deep ocean has functioned across geological time as a kind of parallel biosphere running its own evolutionary clock at a vastly slower speed, sheltering designs that the surface world discarded hundreds of millions of years ago. If that is true, the frilled shark is one data point in a larger argument that the ocean floor is not an empty museum. It is a working archive. From the deep Pacific, we rise toward the surface and then descend underground into the tropical leaf litter of South America, where an animal that has no business being soft-bodied has been walking essentially unchanged since the Cambrian. The velvet worm. 538 million years ago, life on Earth made a decision. In a geological instant, measured in tens of millions of years, the basic body plans of nearly every animal alive today appeared in the fossil record simultaneously. Shells, eyes, limbs, segmentation, the concept of a head. This was the Cambrian explosion, and it was the most consequential biological event in the history of complex life. Most of the designs it produced failed. The ones that survived either evolved beyond recognition or disappeared. Except one, which crawled out of the Cambrian, walked into the nearest patch of moist leaf litter, and has been doing essentially the same thing ever since. The velvet worm, a member of the phylum Onychophora, looks like a caterpillar designed by someone who had only heard caterpillars described secondhand. Its body is a soft, segmented tube, typically between 1 and 15 centimeters long, covered in hundreds of tiny papillae that give it a velvety texture. It moves on between 14 and 43 pairs of stumpy, unjointed legs called lobopods, without the precision of an insect or the muscle coordination of a vertebrate, just a slow, deliberate shuffle through the dark. Its fossil relatives appear in the Burgess Shale of British Columbia, Canada, dated to approximately 508 million years ago, in deposits described extensively by paleontologist Charles Walcott beginning in 1909. Researcher Simon Conway Morris at Cambridge reinterpreted several Burgess Shale fossils in the 1970s and 80s, recognizing Cambrian lobopodians as relatives of the living onychophorans. The morphology of the 508 million-year-old fossil and the morphology of the animal living in the rainforest floor today are, in all meaningful ways, the same. What makes this remarkable beyond the age alone is what the velvet worm is made of. It has no hard parts, no shell, no exoskeleton, no internal skeleton of any kind. It is a tube of soft tissue with stubby legs. Soft-bodied organisms almost never fossilize. Their remains decay before mineralization can occur. The Burgess Shale preserved them only because of a specific geological accident, an ancient mudslide that buried organisms in anoxic conditions before bacterial decomposition could proceed. For a soft animal to appear in the Cambrian record at all required a geological miracle. For that same soft animal to still be alive today, walking across a leaf in the Amazon basin or the forests of South Africa or the rainforests of Southeast Asia is something the standard models of extinction vulnerability do not cleanly account for. The velvet worm is classified as a sister phylum to the arthropods. It shares a common ancestor with insects, spiders, crustaceans, and every other jointed limb animal on Earth. But it diverged from that lineage before the arthropod exoskeleton evolved. It is a living snapshot of what the ancestor of all arthropods looked like before it hardened into something we would recognize. It breathes through tracheae, air tubes penetrating the body wall, a system shared with insects, suggesting this respiratory design was established in the Cambrian and never revised. Its hunting mechanism is equally ancient and equally strange. The velvet worm possesses two oral tubes flanking its mouth that fire a sticky protein-based adhesive, coating prey in a biological net that immobilizes insects, spiders, and small millipedes from distances of up to 30 centimeters with surprising precision. This slime jet system has no analog in any other living animal group. Some researchers have identified structures in Cambrian fossils consistent with a similar mechanism, which would mean this hunting strategy has been operational for roughly 500 million years. The velvet worm's global distribution across South America, Southern Africa, Southeast Asia, and Australasia mirrors the ancient supercontinent Gondwana. The phylum was already globally distributed before Gondwana began breaking apart approximately 180 million years ago. Populations were carried apart by continental drift, not dispersal, which means these separate lineages have been evolving in isolation since before the Cretaceous. Biologists explain this survival through ecological specialization in stable microhabitats. Moist leaf litter and rotting logs in tropical forests have maintained roughly similar conditions since the Carboniferous. The slime system is efficient enough that no body plan modification was required. The explanation is reasonable, but it does not resolve the Permian extinction, during which an estimated 70% of terrestrial species were eliminated even from tropical environments. The velvet worm had no armor, no burrowing ability, no dormancy mechanism, no obvious tolerance for the temperature spikes and oxygen depletion that characterized the Great Dying. By every measure of extinction vulnerability, it should have been among the first to go. Some evolutionary biologists have proposed that the answer lies in a concept they call ecological invisibility. The velvet worm occupies such a narrow, deep microhabitat that extinction events operating at the ecosystem level simply never reached it. The catastrophe passed overhead, while the velvet worm continued shuffling through the dark beneath a log. If that is true, it suggests something uncomfortable. That some of the most ancient lineages on Earth are persisting not because of exceptional resilience, but because they are so small, so hidden, and so far below the surface of ecological notice that disaster has never bothered to look for them. From an animal that hides in the dark under leaves, we move to an animal that hid from science for 130 million years and was only discovered because it came up for air. The purple frog. For 11 months of the year, it does not exist, not in any way you could observe. It lives entirely underground, somewhere beneath the soil of the Western Ghats Mountains in the state of Kerala, in southwestern India. It feeds on termites through a narrow, pointed snout. It has never seen sunlight. It has never been photographed. It has never been documented in any natural history survey, despite the Western Ghats being one of the most intensively studied biodiversity hotspots in the world, surveyed by botanists, herpetologists, and naturalists for centuries. Then for roughly 2 weeks each year, triggered by the pre-monsoon rains, it surfaces. It is approximately 7 centimeters long. It is a vivid, unmistakable purple. And in October of 2003, it came up for air at exactly the wrong time, within reach of a net held by biologist S.D. Biju. Biju and colleague Frankie Bosui formally described the purple frog, Nasicobatrachus sayadrensis, in a paper published in Nature in 2003. The paper's reception in herpetology was immediate and significant. Here was a species not merely new to science, but belonging to a family previously known only from the Seychelles Islands, located over 3,000 kilometers away across the Indian Ocean. Not a close relative living on a neighboring continent. The same family. And the molecular clock, calibrated against known divergence events, placed the split between the Indian and Seychelles lineages at approximately 130 million years ago, in the Cretaceous period, when India was still an island drifting across the Tethys Ocean. India's separation from Gondwana began approximately 130 million years ago. The subcontinent then spent roughly 80 million years in geographic isolation, traveling more than 9,000 kilometers before its eventual collision with Asia, carrying its biological passengers with it like a biological ark. On that ark was a frog. Small, purple, termite-eating frog whose lineage had already been separated from all other frogs for over 100 million years, drifting across an ocean on a moving continent, breeding in its burrows, surfacing for 2 weeks every monsoon, leaving no trace visible from the surface, surviving the KPG extinction, surviving every subsequent event that reshaped the subcontinent, completely undetected by modern science until 2003. The animal's morphology reflects its underground life with a thoroughness that suggests deep evolutionary commitment. The body is bloated and rounded. The head is tiny and conical, disproportionately small relative to the body, with a narrow snout adapted for pushing through soil and accessing termite tunnels too small for any other predator. The eyes are small. The hind legs are built for digging rather than jumping. It does not look like what most people think a frog looks like. Described in print, it sounds implausible. Its mating call is produced while the male floats briefly at the water surface during the 2-week emergence window, and acoustic analysis has shown that the call structure is unlike any other frog species in India, but shares characteristics with the calls of its zooglossid relatives in the Seychelles, separated by 3,000 kilometers of ocean and 130 million years of independent evolution. By 2017, genetic surveys in unexplored Western Ghats terrain had already identified at least one additional undescribed species within the same lineage. The population we know about appears to represent only part of a larger hidden radiation. Mainstream biogeographers explain all of this through Gondwanan vicariance, the passive separation of once continuous populations by continental breakup, a well-documented mechanism that explains distribution patterns across the southern hemisphere. The fossorial lifestyle is understood as a response to the seasonal aridity of the Deccan Plateau, a practical adaptation to months of inhospitable surface conditions. The late discovery is attributed simply to the animal's behavior. It was always there. No one looked underground at the right moment. But some evolutionary biologists ask a harder question. 130 million years of island isolation on a subcontinent that underwent enormous geological and climatic upheaval during its drift, normally produces dramatic divergence. The purple frog's lineage shows remarkable conservatism relative to that expectation. And the fact that a 7-centimeter unmistakably purple frog that surfaces for 2 weeks every year could avoid scientific detection in one of the world's most surveyed biodiversity hotspots until 2003 should, at minimum, adjust our confidence in what else we might be missing. From an animal that hid beneath the soil of a drifting continent, we move to the open ocean, where a creature that has existed since before the dinosaurs is still performing a function that no other living animal has managed to replicate. The Nautilus 500 million years ago, the Nautilus was the apex predator of the ocean. It ruled the seas before fish had jaws, before anything lived on land, before the concept of a spine had been tested by evolution. Its relatives, the ammonites, eventually became one of the most successful groups in the history of marine life, diversifying into an estimated 10,000 species across 350 million years, filling every ocean on Earth with coiled, chambered shells of every conceivable size and geometry. And then, 66 million years ago, at the KPG boundary, every single ammonite on Earth died. Not most of them, all of them. Not one ammonite species survived. The Nautilus, which shared the same basic body plan, the same coiled chamber system, the same ancient oceanic lineage, did not die. It kept swimming. It is still swimming today. And the reason one survived while the other was completely eliminated remains, to a degree that surprises researchers who study it, genuinely unresolved. The earliest nautiloid fossils date to the late Cambrian, approximately 500 million years ago, making the Nautilus lineage contemporaneous with the horseshoe crab as one of the oldest continuously surviving animal designs on Earth. The shell is a logarithmic spiral, each chamber maintaining a mathematically precise ratio to the next as the animal grows. The siphuncle, a tube running through every chamber of the shell, allows the animal to regulate buoyancy by adjusting the ratio of gas to fluid in each compartment. This system has been documented continuously in the fossil record from the Cambrian to the present without fundamental alteration. The Nautilus has between 64 and 90 tentacles depending on species, none with suckers, a primitive configuration absent in all modern cephalopods such as octopus, squid, and cuttlefish, which evolved suckers as a later refinement. The Nautilus's eyes have no lens. They function like a pinhole camera, open to the water, forming dim images without the optical precision of the vertebrate eye or the compound eyes of insects. Everything about its sensory and anatomical toolkit is ancient. Researcher Peter Ward at the University of Washington has spent decades studying living nautiluses. In 1984, Ward rediscovered the second living genus, Alanautilus scrobiculatus, off Papua New Guinea, after it had not been observed for 70 years. He noted that Alanautilus carries a hairy, mucus-coated shell found in no other living cephalopod. The current IUCN assessment lists nautiluses as vulnerable to near threatened, primarily due to collection for the ornamental shell trade. A lineage that survived 5 mass extinctions is now threatened by souvenir markets. Paleontologist Neil Landman and colleagues at the American Museum of Natural History have studied the ammonite nautilus survival disparity extensively. The leading hypothesis involves eggs. Nautiluses lay large, yolk-rich eggs that can sustain developing embryos for months without any external food source. Ammonites are thought to have produced small plankton-dependent larvae that required immediate access to surface productivity to survive. The KPG impact winter collapsed surface photosynthesis globally for an estimated 10 years. Ammonite larvae, dependent on surface plankton from the moment of hatching, would have starved in the first generation. Nautilus embryos, sealed inside large yolky eggs in deeper water, could have developed through the entire impact winter without ever needing to feed at the surface. It is an elegant explanation, but it raises its own question. Ammonites diversified into 10,000 species across 350 million years of evolutionary experimentation. In that time, with that diversity, not one ammonite lineage ever independently evolved the large, deep-water egg strategy that saved the Nautilus. Not one out of 10,000. Some researchers find that statistical gap harder to close than the standard model suggests. The Nautilus lives at depths between roughly 200 and 700 meters during the day and ascends to shallower water at night to feed, a behavior called diel vertical migration. It navigates this daily depth change using its buoyancy system, a solution to the problem of moving through the water column that was perfected 500 million years ago and has not been revised since. Everything we have looked at so far has survived by hiding in deep water, underground, on remote islands, in hidden canyons. The last animal on this list survived in plain sight on every continent, studied by science for over 2 centuries, and it may be the most biologically impossible thing alive. The platypus. In 1799, a dried specimen arrived at the Natural History Museum in London from the new British colony of Australia. The scientists who examined it were certain it was a fraud. They took scissors to the bill, physically searching the seam where someone must have sewn a duck's beak onto a beaver's body. There were no stitches. The bill was real. The animal was real. And as the decades of study accumulated, it became clear that this was not simply a strange mammal. It was a mammal that appeared to have borrowed components from at least 3 separate evolutionary lineages and assembled them into something that, by any conventional model of biology, should not function. The platypus, Ornithorhynchus anatinus, belongs to the order Monotremata, the most ancient surviving mammal lineage which diverged from the Therian mammals, meaning all placental and marsupial mammals, approximately 166 million years ago in the late Jurassic period. This divergence was confirmed and dated by a genomic consortium led by researcher Wesley Warren at Washington University, published in Nature in 2008. In the 166 million years since that split, Therian mammals diversified into over 5,000 species, from blue whales to bats to humans. The monotrem lineage, in the same time span, produced exactly 5 living species, 4 echidna species, and the platypus. Evolution kept offering the monotremes new directions. They declined them all. The platypus lays eggs, a retention from the amniote ancestor shared between reptiles and mammals before the Therian lineage abandoned egg-laying. This was confirmed definitively by zoologist William Caldwell in 1884, who telegraphed the news from Australia to a scientific meeting in London in 4 words that stunned Victorian biology. The eggs are small, leathery, and incubated against the mother's body. The young that hatch from them then drink milk, but the platypus has no nipples. Milk seeps through patches of specialized skin on the mother's belly, and the young lap it from fur. The lactation genes in the platypus genome include sequences with structural similarity to reptilian egg yolk proteins, a direct molecular bridge between reptilian and mammalian reproduction preserved in a single living animal. The bill contains approximately 40,000 electroreceptors and 60,000 mechanoreceptors. When the platypus hunts, it closes its eyes, its ears, and its nostrils, and navigates and locates prey entirely through the electrical fields generated by the muscular contractions of aquatic invertebrates. No other mammal on Earth possesses electroreception. The platypus developed in its bill a sensory system that the entire rest of mammalian evolution never arrived at independently. Males carry venomous spurs on their hind ankles. The venom contains defensin-like peptides structurally related to compounds found in reptile and snake venoms, not related to any known mammalian toxin. Venom production appears to have evolved independently within the platypus lineage, not inherited from a common venomous mammal ancestor. The 2008 Warren et al. Genome paper delivered one more surprise. The platypus has 10 sex chromosomes. Males carry 5 X and 5 Y chromosomes. All other placental mammals carry 2. The arrangement shares partial homology with bird sex chromosomes, not mammal sex chromosomes, suggesting the platypus's sex determination system is a reptilian inheritance that therian mammals replaced and monotremes retained. And the platypus has no functional stomach, no acid-producing region, no pepsinogen genes. The Warren team confirmed these genes have been deleted from the genome entirely. Food passes directly from esophagus to intestine. At some point in the 166 million year monotreme lineage, the stomach was lost and the genetic instructions to build one were permanently erased. Evolutionary biologists explain all of this as phylogenetic relic status. The platypus preserved ancestral features common to the mammal-reptile transition, because the monotreme lineage split off before the full mammalian toolkit was assembled. Egg-laying, electroreception, and the 10-chromosome sex system are retained or modified ancestral conditions. The missing stomach is a secondary loss, common in dietary specialists. These are serious arguments backed by molecular evidence. But the Warren team's genome analysis also identified immune system gene families in the platypus that are not found in any other living mammal, ancient lineages that therian mammals appear to have discarded during their own evolution. Researcher Catherine Belov at the University of Sydney described the platypus immune system as similarly archaic to the rest of its biology, an archive of molecular tools that the rest of mammalian evolution moved away from. The platypus did not fail to assemble a modern mammal. It assembled something else, something older, and then survived 6 mass extinctions with it. The oldest confirmed platypus fossil, a species called Monotrematum sudamericanum, was found in Patagonia, Argentina, dated to approximately 61 million years ago, in the Paleocene, confirming that the monotreme lineage once extended to South America. The lineage has been contracting geographically for 60 million years, retreating to Australia and Tasmania, getting smaller, losing species, losing continents. And yet the template at the center of it, this impossible combination of reptilian inheritance and mammalian innovation, electroreception and egg-laying and blue venomous spurs and 10 chromosomes and no stomach, is still here. 10 animals. 500 million years of survival between them. 5 mass extinctions crossed, some of those crossings made multiple times by the same lineage. The coelacanth survived an impact that killed 75% of all species and then hid so completely it left no fossil trace for 66 million years. The horseshoe crab walked through 4 separate apocalypses without revising its body plan, and its blood is now the safety standard for every vaccine on Earth. Stromatolites built the atmosphere, caused the first mass extinction, and then survived 4 more. The tuatara carries a third eye, inherited from a 240 million year old ancestor we do not fully understand. The Wollemi pine cloned itself in a hidden canyon for 2 million years after disappearing from the fossil record and was found by a park ranger with a rope. The frilled shark has been hunting in the dark with Cretaceous teeth for 80 million years and produces the slowest developing young of any vertebrate alive. The velvet worm has walked through 5 catastrophes on the same soft, unarmored body it has used since the Cambrian. The purple frog drifted across an ocean on a moving continent for 130 million years and escaped scientific notice until 2003. The nautilus watched its 10,000 species family go extinct at the KPG boundary and kept swimming. And the platypus is a mammal that lays eggs, has no nipples, no stomach, no suckers, 10 sex chromosomes, a bill full of electroreceptors, and venom borrowed from reptile chemistry, and has been doing all of this for 166 million years. The standard scientific explanations for each of these animals are, taken individually, reasonable. Deep-sea refuges, stable microhabitats, island isolation, reproductive strategies, ecological invisibility. Presented case by case, each makes sense. But the pattern they form together is harder to dismiss. Every single one of these animals required its own unique exception to survive. Each one found a different door that evolution and extinction somehow failed to close. If our models of extinction were as complete as we believe, why does every impossible survivor require a custom exemption? The mainstream view is that these are simply the statistical outliers that any sufficiently long timeline will produce. Given enough time, given enough species, some lineages will find the combination of traits and circumstances that carries them through. The alternative view, held by a smaller number of researchers but not an incoherent one, is that these animals represent a different kind of evidence. Not outliers in a well-understood system, but indicators that the system is not as well understood as we present it. The biological resilience operates through mechanisms we have not yet fully characterized. That the deep ocean, the underground, the hidden canyon, the drifting continent are not just refuges. They are archives. And what they have preserved across timescales that dwarf human civilization by factors we struggle to hold in our minds is a series of questions that the textbook answers have not yet closed. Most people walk past a horseshoe crab at the beach and see a curiosity. They see a nautilus shell in a gift shop and buy it as decor. They hear that the platypus lays eggs and file it under weird nature facts and move on. You stayed, which means you felt the weight of what these animals actually represent. Each one is a data point in an argument that the story of life is older, stranger, and far less resolved than we are taught to believe. This channel exists for people who are not satisfied with the footnote. Hit subscribe and stay in the archive. And drop a comment telling me which animal on this list you think is the hardest to explain away. Make your case. I want to see the reasoning. 500 million years ago, the ancestors of some of these animals were already ancient. Continents have moved, oceans have opened and closed, the sky has been darkened by ash and impact winter not once but 5 times. And through all of it, something kept swimming, kept burrowing, kept pressing its biology against the dark and refusing to stop. The coelacanth is still moving those lobe fins in the deep water. The stromatolites are still adding their fraction of a millimeter to a column that was old before animals existed. The platypus is still hunting by electricity in an Australian river with a body that should not work, working perfectly. The rules say they should be gone. They are not, and that may be the oldest truth the natural world has ever told us. Survival does not follow rules, it just keeps going. Subscribe, because the archive runs deeper than you think.